What the hell's going on around here? Okay, there it is here. So try to go off cam, maybe that'll help. Do I not have an audio avatar? I guess it doesn't matter that much. Okay, so just wanted to share this. Just gonna do more with it, but I don't have a lot of time right now. Close to winding down time here at the house. I wanted to read from this article. I was in Missing Mod's stream for a minute earlier. So hope citations down in the description. This is a little bit older PubMed article. The origin and evolution of the surfactant system in fish. Insights into the evolution of lungs and swim bladders. We'll probably talk more about this at a later date, but just kind of want to read through this article. Humans being fish and some such nonsense. People who don't really understand how evolution works Okay, so we'll just read it straight through the text here. Abstract. Several times throughout their radiation, fish have evolved either lungs and swim bladders have to see, or as an accessory respiratory organ. Therefore, the presence of air-filled bladders or lungs in different groups of fishes is an example of convergent evolution. We propose that air breathing could not occur without the presence of a surfactant in epithelial cells lining the pharynx. Here we present new data on the surfactant system in swim bladders of 3 teleus fish, the air-breathing I'm going to mispronounce this, Pyruku Aerops cyprinoids, and the non-air Aratus. We determine the mechanical by homologies. We relate the presence and structure of the surfactants of the other members, Osteotes, Actinopterygi. Snapper and tarpon swim bladders are lined with squamous and cuboidal epithelial cells, respectively, containing membrane-bound phosphated choline phospholipid PL profile of lavage material surfactant Lyra, Ruku, and tarpon. Lamellar bodies in tarpon and snapper, SPB in tarpon, and Pyruku lavage, and SPs, A, B, and D in swim bladder tissue of the tarpon provides strong evidence that the surfactant system of other fish and of tetrapods. This study is the first demonstration of the presence of SPD in the air-breathing organs of non-ruminant actinopterygian fishes. The extremely high cholesterol PL ratios of surfactant extracted from Pyruku bladders are characteristics of the surfactant system. Not scrolling. Despite the osteotheses, their surfactant is uniform in composition and may represent the vertebrate protosurfactant. PubMed disclaimer. This site can't be reached. It's a Google search. Okay, here we go. These are lungfish. Citation fucking needed. You're just moving like really, really slowly. It's not popping. Okay, well, let me just do this. The 6 species of lungfish possess both lungs and gills and are the closest extant relatives of tetrapods. Here we report a single cell transcriptome atlas of the West African lungfish. This species manifests the most extreme form of terrestrialization, a life history strategy to survive dry periods that can last for years, characterized by dormancy and reversible adaptive changes of the gills and lungs. Our atlas highlights the cell type diversity with phenotype changes of terrestrialization of fishes, as well as of the West African lungfish. The atlas here provides a valuable resource for further exploration of the respiratory system evolution in vertebrates. The water-to-land transition of vertebrates is a landmark event sequencing to explore the adaptations in African lungfish lungs and gills to terrestrialization. Introduction. The water-to-land transition of vertebrates, or terrestrialization, is a landmark event in evolutionary history that necessitated various adaptations to allow exchange via bladder allows aerial and molecular data suggest the lung and swim bladder are homologous ancestor by an air-breathing holosty fishes, the bowfin and the gars, that was further repurposed by teleost fishes to facilitate buoyancy and, in some species, act as a sensory organ. Lungfishes, a group of lobe-finned fishes, emerged around 400 million years ago and are the closest living relatives of tetrapods. While it is appreciated that extant lungfish species have diverged in various ways, some possess biome for air breathing. Understanding the respiratory condition promises to reveal insights into fundamental adaptations associated with early tetrapod regenera. The Australian lungfish has a single lung, while the South American lungfish and 4 African species have lungfish are obligatory air breathers. They depend on lung respiration. In other words, their gills may be more important for as observed. While the Australian lungfish respires naturally during short-term courtship behavior, toxic water conditions. Lungfishes is dormancy during the dry period. They can last for months. In this hostile environment, they cease food to low levels. And these lungfishes rely on estivation, but others argue that exception does not downregulate its metabolism. We will here employ the term terrestrialization coined by Perry lungfishes. And Schürch S resides in a burrow to counteract water loss. Molecular technologic the date of aquatic and lung transport and may not reliably or comprehensively capture the expressed gene set of a species if the genome is very large or complex. Moreover, single-cell RNA sequencing technologies now enable the full cellular diversity of organs to be characterized. 2 recent studies succeeded in sequencing, assembling, and annotating the genomes of the West African and Australian lungfishes. I think that's something mathematical or some shit. With 2 lungfish reference genomes in hand, it becomes possible to more systematically profile lungfish organs. Here we use single-cell RNA sequencing to profile the transcriptional landscape of the lung and gill of the West African lungfish in aquatic and terrestrial environments. To validate cell type specific genes, we employ in situ hybridization. We also perform cross-species analysis to investigate the evolutionary conservation and heterogeneity of the West African lungfish lung and gill compared to fully terrestrial and aquatic species. Results. A West African lungfish lung and gill cell type atlas. To study cell type diversity, we generated scRNA sequence data of lungs and gills from 3 West African lungfishes maintained under terrestrialized conditions for 33 days and 3 lung or 6 gill individuals from fresh water. We obtained 53,605 and 87,347 cells from 21 lung and 27 gill scRNA sequence libraries, respectively, and detected more than 30,000 feature genes. The number of captured genes in cells is comparable to a bulk RNA sequence study of the East African lungfish lung, a published single-cell datasets of other aquatic species. And this is describing each figure shown here. You can look at it yourself and link the article. We applied Surfactant to identify marker genes and predict cell clusters and employed the unsupervised dimensionality reduction method, or UMAP, to visualize the clusters. We identified 14 lung cell types and 22 gill cell types. We also compared the lung and gill cell type repertoires, revealing broad cell type similarities, as well as organ-specific cell types, such as alveolar cells in the lungs. We next focused on cell type annotation and their marker genes. In the lung, our analysis revealed epithelial cells, stromal cells, endothelial cells, and immune cells, with a large number of alveolar cells. Selected marker genes are shown in supplementary figure 1C. Our cell type classification is consistent with an electron microscope study of the marbled lungfish, showing obvious alveolar epithelium cells, elastic tissue, stromal cells, and blood vessels. We identified alveolar epithelium cells in our data using the gene SLC34A2, which has a phosphate transport function and is highly expressed in the apical membrane of human type 2 alveolar epithelium cells, or AT2 cells. Interestingly, morphometric and single-cell transcriptome appraisals of mammalian lungs suggest that they have a much smaller proportion of alveolar epithelial cells, by about 20%, but additional alveolar and immune cell subtypes, about 40 to 60%. Terrestrial animals harbor various alveolar cell types with canonical marker genes, while expression patterns in our scRNA sequence data and electron microscopy of the marbled lungfish did not effectively stratify further alveolar epithelium cell subtypes. Similarly, a recent cell atlas of a xenopus lung identified a single alveolar epithelium cell type, the stromal marker decorin, DCN, an extracellular matrix protein was used to classify stromal cells. The gene ARPC1B, which plays a role in inflammatory diseases and has macrophage-specific expression in human GTEX. Data analyzed by the Human Protein Atlas showed high expression levels in lung macrophages. We classified other cell clusters using general markers of erythroid cells, HBB and HBA2, vascular endothelial cells, e.g. FL7 and VWF, epithelial cells other than alveolar epithelium, KRT8, KRT17, and GSTA1, and lymphoid cells, SH2D1A. We also identified various cell types in the gills of the West African lungfish. Selected marker genes are shown in supplementary figure 1D. The number of immune cells in the gill under freshwater and terrestrialization conditions is more than that of the lungs, likely because that its external gills are in direct contact with water and continuously exposed to toxins and pathogens. We identified major lymphoid cell types in the West African lungfish gill, including T cell, CD8, and T cell, B cell, and NK-like cell. We found the NK-like cell cluster expressed representative markers of mammal granzyme M, GZMM, and granzyme A, GZMA, as well as genes denoted NK cell type specific in the molecular structures in the molecular signatures database, MSIG-DB, supplementary data. Rare cell types identified included platelet cells, which show high expression of TIMP4, major inhibitors of matrix metalloproteinases in human platelets, and F13A1, coagulation factor, A1 polypeptide. We identified two types of ionocyte cells and ionocyte DMRT2 cells that regulate salt balance and show environment-induced gene expression plasticity of ion transporters, agreeing with West African lungfish scanning electron and immunofluorescence micrographs. To validate the cell types classified using marker genes, we performed H&E staining and RNA fluorescence in situ hybridization on cross-sections of West African lungfish lungs and gills. The fish results were consistent with the physiological structure of the marbled lungfish lung, the mammalian lung, and gill filaments of ray-finned fishes. Moreover, gene set enrichment analysis, or GSEA, of genes differentially expressed by cell types consistent with cell type classifications. Cellular and molecular features of lungfish lungs during terrestrialization. Lungfish terrestrialization observed in the South American lungfish and African lungfishes is an episodic response to the extreme environment during dry periods and are reversible adaptive changes of the gills and lungs. A period of dormancy, the maintenance phase, which can last for years, then follows. While the West African lungfish is an obligate air breather, its respiratory organs nevertheless show fundamental morphological changes during the maintenance phase. The paired lungs become expanded and better vascularized. We performed scRNA sequence after 33 days at the maintenance stage, where the West African lungfish exhibit a lower oxygen consumption and respiratory rate, and compared its expression to fish maintained in fresh water. We focused primarily on the lungs since its comparatively similar structure during freshwater conditions should allow for more biologically relevant gene expression comparisons. Few discernible differences in lung and gill cell types were detected after 33 days at the maintenance stage compared to freshwater lungfish. To further characterize molecular changes in cell types during West African lungfish terrestrialization, we performed differential gene expression analysis and observed more downregulated DEGs in 14 cell types of the lung and 16 gill cell types. In agreement with a lowered respiratory rate during terrestrialization, erythroid cells, or red blood cells, showed reduced expression of the aminolevulinate synthase isoform genes, ALICE1 and ALICE2, enzymes that catalyzes the heme biosynthetic pathway in both the lung and gill after 33 days at the maintenance stage. In the lung, I don't know why I keep having such problem with this word, alveolar epithelium plays a fundamental and essential role in gas exchange. In this cell type, we identified 328 upregulated and 639 downregulated DEGs, genes encoding the electron transport chain essential for cellular energy production were downregulated, likely reflecting a reduced metabolic rate despite a larger alveolar epithelium cell number. Other downregulated genes of interest include the solute carrier proteins SLC34A2 and SLC22A3. Lung alveolar epithelial genes upregulated in terrestrialized lungfish included NPC2, that's MPC intracellular cholesterol transporter 2, which has a role in lung lipid metabolism and is associated with Niemann-Pick disease characterized by alveolar proteinosis. And the antioxidant enzyme genes MGST1, that's microsomal glutathione S transferase 1, and PRDX1, peroxyredoxin, horribly mispronounced because I'm not a biologist, increased alveolar epithelium expression of antioxidant genes expands on a previous biochemical study showing an enhanced antioxidant capacity of the Nigerian lungfish heart and brain during terrestrialization. Genes such as DNA JB13, CCDC39, and DNAAF1 were upregulated in ciliated cells and likely reflect the expanded lung under terrestrialization. genes upregulated by lung macrophages during terrestrialization were enriched for the endocytosis and phagocytosis pathways. Genes of interest include APOE, apolipoprotein E, and MARCO, or macrophage receptor, with collagenous structure. APO is secreted from macrophages and plays an important role in immunoregulation. MARCO is a scavenger receptor that plays a crucial role in the immune response against microbial infections. The vascularized lungfish lungs downregulated VWF, von Willebrand factor, and SELP, or P-selectin, in endothelial cells, which may reflect a reduced blood clotting ability to prevent intravascular thrombosis. As previously reported, for the terrestrialized West African lungfish liver. And once again, you can kind of see the results of the charts that are made here. I'll make this all available. We next considered cellular crosstalk between different cell types in freshwater and during terrestrialization using cell phone DB. I like how the cell phone DB, a database of ligand and receptor interactions that considers their subunit architecture. We detected 35 significant ligand receptor pairs, or LR pairs, among the 14 lung cell types. The freshwater lungfish showed more connections among different cell types in the lung. 31 LR pairs, 15 shared interactions with terrestrial as lungfish. To better understand the cell type interactions, we perform geo-enrichment analysis on ligand receptor pair genes from the lung, revealing enrichment for blood vessel formation. We considered LR pairs with differential gene expression during terrestrialization and identified 2 pairs with changes in the lungs, NRP1, A. The transmembrane glycoprotein neuropilin 1, NRP1, is a co-receptor of vascular endothelial growth factor, A, VEGF-A, and semaphorin 3A. Binding of NRP1 on the surface of endothelial cells by VEGF-A stimulates vascularization via vasculogenesis, and NGMA3A inhibits vasculogenesis. VEGF-A and NPR1 show lung-specific expression in species ranging from basal rayfin fishes to lobefin fishes and tetrapods. Interestingly, sarcopterygians, NRP1 loci share a candidate enhancer speculated to facilitate the vertebrate water-to-land transition. While further studies are needed to validate signaling interactions between West African lungfish cell types, in particular the contribution of non-epithelial cell ligands to vascularization mediated by endothelial cells, we observed that the freshwater lung showed higher expressions of NRP1 VEGF-A LR pairs. This included a higher expression of NPR1 in endothelium and higher expression of VEGF-A in alveolar cells of the lung. The expression level of SEMA3A was to conclude an antagonistic role in the West African lungfish. However, the previous study found that the lung of terrestrialized West African lungfish are better vascularized and expanded, hinting that SEMA3A outcompetes VEGF-A vascularization effects under freshwater conditions, despite its high gene expression. Difference in gills between freshwater and terrestrialized condition. African lungfishes are oxygen via their lungs, while 10% is acquired via the gills and skin. The manifestation of terrestrialization in the two respiratory organs are vastly different, forms an immunoprotective cocoon from mucus secreted by its gills. Thus, its gills are round. Nevertheless, most of the carbon dioxide is released via ionocytes of the fish gill epithelium in fresh water. Carbon dioxide release organ. During terrestrialization, lungfish gills regress and become covered in mucus, and the lung takes over the carbon dioxide release role. Mitochondria-rich ionocyte cells play a dominant role in osmotic and acid-based regulation. After 33 days of the maintenance phase, we identified downregulated DHMRT2 cells respectively. Of these DHEs, 867 were shared by the two cell types and showed enrichment for mitochondrial localization. The 685 to 1,552 DHLs were enriched for biological processes associated with ion transport, in agreement with the gill osmoregulatory function. Given the various types of immune cells in the West African lungfish gill, we grouped them using the human classification system into myeloid, dendritic cells and macrophages, and lymphoid, i.e. T cells and NK cells and other lymphoid cell lineages. Myeloid cells are part of the innate immune system, while lymphoid cells form part of the adaptive immune response. We observe relatively few myeloid cells. However, these cells are shorter lived than most lymphoid cells, and our single sampling point, 33 days, may not have adequately captured their cell dynamics at the maintenance phase. We observed that many glycoproteins or factors showed lower expression in most immune cells of the gill at the maintenance phase. However, genes such as CXCL2, PMP2, and ABCC9 were highly expressed in myeloid cells of terrestrialized gills, in accordance with the roles of terrestrialization. In proliferative cells, ostensibly a proliferative lymphoid cells with enzymes and genes associated with mitochondrial bioenergetics. We also considered 51 significant ligand receptor pairs, or LR pairs, showed more connections than terrestrialized geoanalysis of total gill ligand receptor pair gene morphogenesis. In contrast, the lung data, the higher expression in terrestrialization gills. This included higher expression of NPR1 in endothelial cells and higher expression of the VEGF-A in stromal cells of the gill. This is consistent with the observation that the vasculature of marbled lungfish is dilated but Celtic and fossil evidence suggests that lungfishes are pivotal integrated are West African lungfish, lung, mouse lung, zebrafish swim bladder, Atlantic salmon gill, and zebrafish gill. We found that epithelial cells and granulocyte cells of the lung and swim bladder show high similarity among these four species, suggesting that these cell types share ancestral cell types. As for the comparison between lungfish and mammals, alveolar epithelium cells, ciliated cells, and lymphoid cells of West African lungfish lungs showed high similarity to mammalian, here human and mouse, AT2 cells, ciliated cells, and T cells. However, many immune cell types in the human or mouse lung show no correlations with the West African lungfish, likely reflecting the distinct immune cell repertoire of these terrestrial animals. In summary, our data indicate broad homology between the swim bladder of rayfin fishes and the lungs of tetrapods and the West African lungfish. Interestingly, the cell type correlations between zebrafish swim bladder and West African lungfish lung are mainly related to innate immune function, and the structural characteristic of human lung cell types are related to air breathing and adaptive immunity. One moment, I have to take this.
Hello?
Hey.
Kevin, it's Melanie.
Yeah, how you doing?
Did you take care of that?
Okay, sorry about that. I got a little bit of crisis averted there. We're good though. Let's see, 9:35. We have a few more minutes. So, how much longer is this article? Scroll through. So I think we can leave it here and conclude tomorrow. Because there's several facets to the thing we're exploring here. But I'm going to leave a link to this if anybody wants to. I was right here, right? Yeah, so we were right about here. So we'll leave it there and continue the study tomorrow. But for now, yeah, thanks for listening. It's been fun. Yo, howdy, lemon pie, dadman walking. Danbury Sports highlights, bad static and breakup in the audio. This has been an ongoing issue. Unfortunately, my earbud went dead just before I started this. Apparently, there's some issue going on with weird, because it usually works just fine. Although I may have adjusted the volume on it earlier. So we'll work on that. But like I said, we'll continue this tomorrow. Thank you all for listening. And I'll drop the link here for the study in side chat and the description before we go. But there's going to be, this is part of an ongoing thing that I'm going to do here. And we'll address, if I can find it, if it hasn't been taken down, the conversation that happened over at Missing Mod's channel earlier, which is kind of the inspiration for this line of inquiry, if you will. So, let's see, edit. Okay, so links for both of the studies will be in the description box, as well as links to all my other socials and music stuffs and recommended pages and stuff like that too. So all right, well, I'll bid y'all adieu. Wish y'all a good night. And be seeing you very shortly. Well, not very shortly, but soon enough or whatever. So be kind, be well, stay trusty, and let's learn something. Learn something new every day. Give it a shot. What do you got to lose? Three minutes? You can do 3 minutes. No, we can't because I deleted the long one. All right. Peace out.
It's a monkey see, monkey do attitude.
And they don't even seem like monkeys anymore. It's like, God damn it. They've been too humanized.
I'm tired of women. I wish I could become one of the gays.
Moist critical? Is that another atheist? Is that more atheism?
Oh my.
No, it's not that hard.
No, there's always, they're so weird, man. You can't figure them out.
I'm really poking the nest or whatever the term is. we got B.S. Lewis.
They won't talk to you directly. They never talk to you directly. You always have to fucking speak in codes with you.
And that is why you read as many books as you can about how to handle women and the psychology. If you want.
Fuck it.
Well then, keep having issues with women.
Mom's busy right now. Those are some memories from the homeschool days.
The problem of Jim Bob solved. Oh, he's just doing silly stuff.
What can I say, Kevin?
If you think you having issues with a woman, just say, why are you having an issue with me?
But the main thing I think of when I think of homeschooling is that it's really not about education.
He put an eggplant on my upper lip, dude. Come on, bro.
It's not really about the schooling. It's about discipleship.
You see, with women,